Hello, this is Mark Isselhart, Maple Specialist with UVM Extension. This presentation talks about sap production when tapping into stained wood, and it explores a little bit about how trees compartmentalize the wounds that can happen occasionally. Tapping into a tree is certainly one of those times, but also a broken branch or some sort of damage to the bark inside of the tree. And we'll also talk about some preliminary data and experiment that I've been working on, trying to understand how much sap we get out of tap holes when we do encounter some stained wood. So most sugar makers, or all sugar makers, know you have to make a new hole every year. And the reason for that is that the tree has a natural process of defending itself against infection. And it's similar to when you or I fall and get a scrape and our body heals up, but it's a little different. It's also a little unique to the anatomy of wood. So we know we have to make a new hole every year, and I'm going to walk you through a little bit about the process of how a tree goes about doing that and prevents the infection and decay from spreading. So specifically, what does tapping do to a tree? And we'll use the illustration on the right to talk about it a little bit. Tapping is a wound, and we are removing a little bit of wood with the drill bit, and we're also removing some wood that basically takes it away from the tree. It's the conversion of wood that is normally conducting water or sap. It's actually being blocked off and compartmentalized. So a portion of the wood, and you can see it in that illustration extending above and below an old tap hole, that wood will no longer conduct sap, or during the summertime when the leaves are on and the tree is photosynthesizing, it won't conduct water anymore. The tree uses this process. It's an active process. The tree uses some of its stored energy to do this, and it's very effective. And sugar maple is really one of the best types of trees at doing this. The tapping intensity, or the number of tap holes you put in a tree, is going to have an impact. So we know that you put one hole in a tree and you make one staining column. If you put two tap holes and you get two staining columns. We know from some previous research that you don't get twice as much sap if you're tapping with vacuum with that added tap. You do get more overall sap, but it's not like the old days when we used buckets. And we could see, if the tree was big enough, you could really see a doubling or potentially even a tripling of the amount of sap you'd get. It's a little different when you start adding vacuum and tubing. So tapping produces an area of non-conductive wood. You could also think of it as stained wood. And diameter growth adds conductive wood. So luckily for the tree, and luckily for maple producers, the tree is each year adding more wood. And the tree will add more wood than other trees if the tree has a little bit of room to grow and the growing conditions during the summer are good. The other thing I'll want to talk about is that this area of stained wood that is the result of tapping, the concept of compartmentalization was really defined by Alex Shigo, researcher for the Forest Service, and he talked about the staining in the concept of walls. So different walls were stronger or more able to prevent the staining from extending throughout the tree. And the walls go from 1 to 4, 1 being the weakest and 4 being the strongest. So let's take a look to see what that really means. Here's an illustration, a drawing from some of Shigo's work. And again, it's meant to illustrate this concept of walls. Now, if you can see where the number 1 is, that's kind of looking down on top of the surface, the cut surface of a piece of wood. And that's really the weakest wall. That's the surface where water and sap flows the most freely, and so it's harder for the tree to shut that down. The next strongest wall is wall number 2, and that represents the later growing season cells. So if you look at a tree in cross section, you'll often see growth rings, and those growth rings represent early wood, which is the cells laid down early in the growing season, and then the thicker walled later season or late wood. Those late wood cells tend to be a little better at walling off infection. So wall number 2 is a little bit stronger than wall number 1. Wall number 3 in this illustration looks like almost like a gray brick wall. That wall is formed by the many ray parenchyma cells that exist that kind of radiate out from the center of the tree. They don't look gray, but this illustration wants to highlight that they are in fact a separate part of the wood anatomy. And that kind of hems in the staining from side to side. It's pretty important when you think about it in terms of tapping, because we see the staining in a tap hole as really only being a little bit wider than the tap hole diameter itself. And that kind of illustrates how strong that third wall is. The fourth wall is actually the new year's growth. So once the season is over, the tree goes dormant. When the tree comes out of dormancy the next spring and starts growing, that new wood is the strongest barrier between infection. So here is a zoomed in picture, a very, very, very magnified picture of some sugar maple wood. If you look at it, for one, it's been stained with an iodine solution to kind of highlight the different parts. What we can see are a couple of different things. Those relatively larger white circles, those are the ends or a segment of some of these large vessels. The vessels are the things that conduct water during the growing season, and they're the things that are conducting sap during the sugaring season. So they're relatively large compared to those other cells that surround them. Those other cells that look kind of like a pale yellow, those are the fibers. Those are the things that give wood its strength. Those are the things that make baseball bats so good, and wood is a good building material. It's really the strength and flexibility that you get with those fibers. So we don't collect sap out of those fibers, even though it looks porous. We're really just collecting sap out of those relatively larger xylem vessels. The last thing I want to point out is the dark lines that you see going up and down. So it might be tempting to think that those are growth rings going side to side, but they're not. They're actually the ray cells. So if you think about those ray cells going up and down, they're actually slightly pie-shaped. They're kind of radiating out from the center. The bottom of your screen, somewhere down there, is the center of the tree, and up at the top of your screen is towards the outside or the bark. And if you know that, then you can kind of look at those cells in between the rays and start to see where the late wood starts to show up. It's a little thicker towards the top. So almost at the bottom of the screen is to the very top is one growing season. So if you remember from the illustration before, wall 1 is the weakest wall that we have in the wood, and that's the one that's hardest for the tree to shut down and close off from infection. Like I said before, the tree does this as an active process. It uses stored energy from previous years, to produce new chemicals and barriers to prevent moisture from moving around the tree. That wall 1, those get formed near the point of the wound, and it happens to plug up or some way wall off those relatively large vessels. So wall 1 forms in those circles, those large vessels. Now the size of the wound is going to impact how many vessels are walled off and how big the staining area is. So that's all related to how big the initial wound is. Wall 2 is, like I said, those relatively thicker late season cells. So at the bottom of your screen, you see that kind of band of thicker cells. And if you go to the top of the screen and look, you can see where that wall 2 would also form above there. I just didn't illustrate it with the red circles. So here's a picture that's a little bit more zoomed out, and you probably would recognize this as an old tap hole that has then been covered over with new growth. And the point of this picture is to show you that, one, if you were to slice right through the old tap hole, you'd see that that wood never really fills in. It just gets covered over by new year's growth. The other thing I want to point out is that the width of the staining is really not that much wider than the width of the tap hole bit itself. And this is where wall 3 forms. So wall 3 forms just on the outside of in a tap hole, just beyond that wall. Now, we don't have good data on what's the impact of tapping just beyond that staining. Chances are you'd get a good amount of sap, but maybe you'd get a little less than you normally would. And there's lots of recommendations and sort of conventions about how far away you should tap from an old tap hole. But we do know, just from segmenting and dissecting many trees and looking at tap holes, that the staining really doesn't extend much further than the old tap hole. And you can see that the tree stimulates new growth, and even more than your regular growth, right around the point of that tap hole. And that's sort of illustrated by that big lump of new tissue that's covering up the old tap hole. In wall 4, now I'm going to turn the tree a little bit, and now we're looking at sort of at the side of the tap hole. This last picture we are looking at from the top down. This is really more looking towards the side of a tap hole. And you can see that, again, this tree has grown for a few years after being tapped. And you can see that staining extends above and below the tap hole, but does not extend any further out into that new tissue. And that's, illustrates how strong that fourth wall is, but it also is good to know that over time, assuming a tree is growing well, you can really tap back in that same area, provided you've accumulated enough new wood to make that possible. And so that's where wall 4, the strongest wall, forms, and it's really the new layer of wood that forms after that infection in the following year. That's where that really strong wall is. Now, certain trees, even within sugar maple, some trees are going to be better. There'll be a little bit of genetic variation. Some trees will be better at walling off tissue and infection than others, and we're still learning quite a bit about the variability and how big those staining columns can be. They can be as little as only 2 or 3 inches above and below, or they can be over a foot above and below the old tap hole. So it can be a challenge. And we also know that over time, that stained wood doesn't go away. It lives on in the tree. And the end of this log, you can see many years worth of tapping, as well as an interaction with the heartwood of the tree. Now you can probably also see that none of these tap holes were cut precisely right through the middle when this log was cut up. So although it has the outline of a tap hole, the actual hole in the tree is either below or above where this point is on the end of the log. And this is just another image to show that staining going vertically above and below the old tap hole, and just to show that it does make a difference. The staining does have a particular orientation, and it also doesn't necessarily follow perfectly in a vertical direction. You can see the tap hole on the left is a little bit tilted to the right, and this is really related to the orientation of the grain under the bark. Sometimes the grain of a tree will kind of tend towards a more of a clockwise orientation, and sometimes it'll be the opposite. And that has a real impact on where that staining will be defined. So all of that kind of leads up to the question that I wanted to try to get at with this experiment, and that was, how much sap do you collect if you were to tap into stained wood? Because when you're out there and you're tapping, luckily you can see some evidence of old tap holes, but some you might not see. Maybe the bark is really rough, or maybe it's snowy out, or maybe you're just in a hurry and not really taking enough time to look to see where those old tap holes are, and you hit some stained wood. And the way you know about it is when you're tapping, the chips should be nice and clean. They should be kind of a blonde or a white colored material, and they shouldn't be brown. They shouldn't be dry, and that would be an indication that you have hit some old stained wood. But with tubing systems, we don't have that individual tree data like we did back with tapping with buckets. We don't know exactly how much sap is coming out of each tree, because that all gets combined into the tubing system. So this experiment was designed to try to figure that out. So relatively small experiment, but we used 20 trees, and they did have a history of tapping. So there was already some staining, some non-conductive wood in the tree, and used 3/16 lateral line material and set it up to generate some vacuum based on gravity. So there was no pump attached to it. Each individual, there was one tree, had one line, and there was one tap on that tree. And of the 20 trees, 10 trees were tapped as you would normally do, to tap into clean wood, but 10 were tapped directly above the previous year's tap hole. So intentionally hitting stained wood. And then we would be able to know with quite a bit of precision how much sap came out of those taps compared to the clean tap holes. So for each of those trees, we collect the total amount of sap and average the sap sweetness. And I was also interested in looking at the actual wood chips, what comes out of that tap hole, and quantifying those a little bit, either how much they each weighs and how much the various percentages, how much was conductive and how much was not. So here's a picture of what it looked like when I tapped directly above an old tap hole. I used a standard 5/16 inch spout, tapped 1.5 inches deep, and went an inch above the previous year's tap hole. And as you can see in this image, it's a little blurry, but the sap was definitely running when I made those holes. An interesting result already was that 3 of the 10 tap holes that I made and then tried to hit stained wood, 3 of the 10 appeared to miss any stained wood whatsoever. In other words, all the chips that came out of the tree were clean. So that was an interesting result that I definitely did not expect, but it does kind of point to the fact that the staining column does not always follow directly above the old tap hole. And again, this is what we'd be looking at in profile, and I would be tapping just an inch above that old tap hole and going through perhaps a little bit of new growth, although this tree has been growing for more than 1 year, but you get the idea. Just to illustrate what the drill bit would be going through, it would be going through some amount of stained wood, some conductive wood, and then some bark. So to collect the wood chips was a bit of a chore, but I was able to rig up a plastic table, essentially just a tray underneath the drill as I was tapping, and collect all the chips, or as many as I could possibly find. And I collected them all, both non-conductive, conductive, and bark all together, put them in a vial and froze them, and then was able to do analysis back in the lab. And this picture shows really what you'd be looking for when you are tapping into clean wood. It's that kind of nice creamy blonde looking chips. They're a little bit moist, but they're not sopping wet, and they're definitely not dry. You can also see a mixture in there of other materials, so bark and inner bark of the tree. So here's a picture of two tap holes worth of wood chips. The one on the left was one that was into a clean tap hole, and the one on the right was into a staining column. And they're both each one is sort of mixed up, so you can see there's a little bit of dark in both. Presumably some of that is bark on the left, and on the right there's a mixture of a little bit of conductive wood as well as a little bit of bark and some conductive wood. So I did spend some time sorting these chips and getting them separated so that I could maybe measure the mass or do some other analysis. And here's an example of one that was tapped into stained wood. So I was able to identify, not with 100% precision, but I think good enough, the parts of the chips that are bark, parts that were conductive wood, and then the relatively larger amount of stained wood or non-conductive wood. And here's an example of one that went right into clean wood. So the outer bark and the inner bark, and then you get into just clean conductive wood. And I was able to measure each of these both in weight but also in sort of relative percentage. So let's get into the results. Again, this was all done just in 1 year, so it's important to remember that years can be different, and we'll be repeating this experiment again and giving more updates next year. So in terms of sap production, when you look at the sap produced from tap holes that were into just clean wood, you can see that data is on the right. Those bars represent the mean amount of sap, and the vertical bar represents the error or the variability. It's a measure of variability. So how much within those clean tap holes, how much could it vary? So some were a little lower, some were a little higher. And then on the left, you can see how much sap or how little sap you get when you do hit that stained wood. And again, those bars represent the mean, and then the vertical dark gray bars represent sort of the variability or how much it could range. So 60% less sap. That was the average. 60% less sap when you hit non-conductive wood. However, that 60% less includes those 3 trees that we did not appear to hit any stained wood, and presumably those were actually clean tap holes, even though they were in the treatment of hitting stained wood. So for the purposes of this analysis, I did it again, or took out those 3 trees from the analysis, which brings the average down even further. So now you're talking a 75% less sap when you hit stained wood. And that's 75% less sap right off the top. That is regardless of how good your tubing system is, how carefully you check for leaks, or how you monitor vacuum, maybe remotely. It doesn't matter how big your vacuum pump is. It doesn't, all those things, how you manage your vacuum pump, if you leave it on or turn it off. This is sap right off the top that just will not come out of that tap hole. And that has potentially big impacts on your total yield and your operations profitability. So something you definitely want to keep an eye on. Now, when we look at the percentage, we have 10 trees of each. So when we look at those stained trees, I'm sorry, before we do that, the sap sweetness, it was interesting to note that the sap sweetness was no different between the trees tapped into clean wood and those tap holes into stained wood. And so that is interesting. Now remember, the stained trees would look a little bit like this, where you'd be going through just a little bit of conductive wood and the rest would be stained. And here's an example again of the relative amount of conductive wood, and you can see pretty clearly that why there's such a big reduction in sap yield. You're cutting into fewer of those vessels. There's less viable tissue for that sap to run through, and therefore you collect less. I was interested to see if there was any pattern between how much staining and how much the yield was within that stain treatment. And there is quite a bit of a relationship, a very strong relationship. The more stained wood you intercept or cut into with your tap, the less sap you're going to get. So you can see when you went through relatively small amount of stained wood, the production was pretty good, and then it drops right off as you go higher and higher in percentage. So what's next? One question we often get is, well, if I hit stained wood, what do I do? Now there's a decision to make. There's either use the tap hole you have or potentially make a second tap hole, but that there are consequences for doing both. If you use the tap hole that you just put in, you'll get quite a bit less sap, but you'll still have only made that one additional staining column. If you put in a second hole, then you'll essentially make another whole column of stain, and there is the potential for a vacuum leak, although we don't have good data on what the magnitude of that would be. And that's one of the things I'd be interested in learning more about, is what's the yield if you do put in a second tap? Do you get back to as if you had hit a clean wood, or is there some reduction based on having that open tap hole somewhere else on the tree? Of course, 1 year of data is just 1 year of data, and it'd be important to do more. So I'd be planning on doing an additional experiment and seeing if the numbers agree from 2 different years. And we will continue that and hopefully have more information in the coming months. So again, this is Mark Isselhart, UVM Maple Specialist, and I'd be happy to answer questions. If you have any, get a hold of me at the UVM Maple Extension website, or you can call the Proctor Maple Research Center. Thank you very much.